the first thing is about lipoproteins. As the name suggests, the lipoprotein, lipid has to be always, it has to be associated with protein to execute some function. That is the main reason behind that is it is completely hydrophobic. So to make it even more hydrophilic, it has to get associated with some protein, and that protein in our body, in this lipoprotein, is apoproteins. Okay, so these apoproteins are usually produced by enterocytes. It's produced by your liver also, and each and every apoprotein will have a different function, and the presence of each apoprotein will tell us what sort of lipoprotein is that actually. So if you look into the lipoprotein score, you have triglycerides, you have cholesterol ester, you have phospholipids, sorry, and free cholesterol. But if you look into the exterior of this thing, as you could see here, this exterior is having a lot of phospholipids. Okay, so these phospholipids are actually going to act as a a communicator between your lipid barrier and your lipid barrier. So apart from that, you could even see small free cholesterol over the surface of all these lipoproteins, and this is what we call as a proper protein. So this is the apoprotein. Okay, so this is the apoprotein, and based on the type of apoprotein, the function of the lipoprotein will vary. We will see about it. So if it is B48, most likely this is going to be chylomicron. Okay, so this is chylomicron. Usually the chylomicron will have the B48 apoprotein, and if it is B100 apoprotein, this assembly and secretion, everything will start from your liver. So this is produced by your enterocyte, whereas the B100 is produced by your liver. Clear? So it is going to produce the B100, and usually the B100 lipoproteins would be VLDL, IDL, and LDL. And of course, we have some special apoprotein called as apoprotein little a. This is going to be the key molecule in coming future. It has gained a lot of momentum in the modern medicine, and we will have to have a separate session on this lipoprotein A, which will be dealt with in a part 2 of this chapter. But grossly, you will have to remember this lipoprotein little a is also produced from your liver, and this is lot more atherogenic. It's going to behave like a LDL, like a small dense LDL, as well as it is going to have some of the plasminogen sort of activity. So it is going to be hyperinflammatory and hyperthrombotic. Okay, so that is the significance of lipoprotein little a. If you look into this, you could see the different type of each and every lipoprotein that are synthesized. If you look into chylomicron, that is synthesized by the intestine. We'll come into that finally. But the basic thing we will have to know, I will just explain it. So when you look into the lipids, the chylomicron will have the highest fat content. Okay. And as the fat content reduces, you could see it's coming, the density slowly increases. That is because protein content would be higher. So this chylomicron has high fat with a subtle protein content. As the fat reduces, then comes your VLDL, IDL, LDL, and finally your HDL. So HDL is going to have a lot of proteins, and it will be very, very dense. So density-wise, HDL stands first, and density-wise, it is very lesser. But diameter-wise, the size-wise, usually the chylomicron is the biggest one. So they can ask simple question: which one would be having the biggest diameter? Which one will be having the highest density? All those questions at that moment you will have to answer. So we will see the fate of chylomicron first of all. So whatever lipids that you are taking, whatever food that you are taking, will be digested by your intestinal lipases. And this digestion generally tends to happen in your proximal intestine. So proximal small intestine, that is where all the pancreatic lipase and all the lipases comes. And finally, the fatty complex food that we eat is broken down. And this is in small pieces. Now, the next important factor that has to happen here is called as micelification, micelle formation. Okay, because these small fat globules has to be further made at least to get more uniform texture, and only then they would be easily taken up by the enterocyte. And for this, bile salts, they play enormous. So once it is micelified, what happens is through the enterocytes, Okay, we have a special transporter called as MP1C1. Through that, all this gets entered into your enterocyte. Within the enterocyte, what happens? These lipids are packed into certain molecules. So this enterocyte will be having endoplasmic reticulum, rough and smooth endoplasmic reticulum, and they both come together, and finally they assist in the formation of chylomicron. Okay, so ApoB48 is produced by the endoplasmic reticulum, and these small lipid particles, that is the triglycerides that you have got, are slowly aggregated, and finally this triglyceride dense lipid particle attached with ApoB48 is now secreted into the lymphatics. So this is the lymphatic. Okay, and from the lymphatics, all these chylomicrons, which are abundant in triglyceride, and to a certain extent, you may also have cholesterol and cholesterol esters. Okay, so cholesterol esters would also be there, and this enters into the lymphatics and down into the thoracic duct, and finally they end up into the venous system. So what happens once inside the venous system? They get acted upon by the endothelial LPL. So that is called as lipoprotein lipase. Okay. So for this ApoB48, it will not be acting on the LPL. It needs some other proteins to facilitate the action of this catalytic activity. They have to have some other protein. So this is donated by your HDL. And HDL will be there in the blood. So what does HDL do? So HDL donates some of its apoprotein. Okay, it donates ApoAB, ApoC, and to a certain extent ApoE. He is a donor. He's a great donor. He will donate all these proteins, apoproteins, to your chylomicron. And now chylomicron receives all these ApoS, ApoA, ApoC, and ApoE. And it's slowly moving in your venous circulation right now. So once it is moving into the venous circulation, what happens is this ApoC and ApoAB both get the attention of lipoprotein lipase. Okay, so ApoC is the main thing which is going to activate the lipoprotein lipase, but ApoAB will also act as a cofactor. So either ApoC is absent or ApoAB is absent or lipoprotein lipase is absent, these chylomicron will not be catabolized at all. Okay, so this is chylomicron, this is ApoAB, ApoC, and ApoE along with ApoB48. So this is the chylomicron. There's a lot of triglycerides and cholesterol esters. Only some level of cholesterol ester it will have. Lot of triglycerides which is coming from the diet is what it has. So what does this lipoprotein lipase do? Automatically it breaks down these triglycerides into free fatty acid and monoacyl glycerol. Lot of free fatty acids gets emanated. Now these free fatty acids, depending on in which tissue is this lipoprotein lipase is present, will be metabolized accordingly. For example, if you see this lipoprotein lipase would be seen in your hepatic, not in hepatic, it would be seen in adipose tissue and in your skeletal muscle. If at all, if this chylomicron is getting into the capillary plexus or the blood plexus of your adipose tissue, this lipolysis will happen and the free fatty acid will be taken up by your adipocyte. Okay, and it will utilize all this in the energy formation, into ATP formation. That is what skeletal muscle does. So finally, this is how a chylomicron, which is digested in your gut, is going through your body and it's finally acting. But I'll come again for a better understanding. I'll give a very simple picture. This is the food that you are taking. The fats in the food are getting acted upon by the lipases in your small intestine and bile salts, and finally misalifications happen. Once they are misalified and they are homogeneously diluted, they gain access into the enterocyte through this particular channel called as MP1C1. Within the enterocyte, from the B48, the apoprotein as well as the proteins are all produced by our endoplasmic reticulum. And finally, all these triglycerides, okay, are all packed up into a chylomicron. This is called as nave chylomicron, very, very nave chylomicron. This is pushed up into the lymphatics, finally into the venous plexus. Once inside the venous system, it receives certain apoproteins from your HDL. All these are donated by a donor HDL. Okay, and once this is called as mature chylomicron. Now, this mature chylomicron, because of the C2 and the AV presence, gets activated by a lipoprotein lipase, and depending on the tissue, it gives all its triglycerides as free fatty acids and glycerol. If it is in skeletal tissue, it is used in energy formation. If it is in adipose tissue, it is stored as triacylglycerides. Okay, so once adequate triglycerides is removed from this chylomicron, the remainder of what it has is called as chylomicron remnant. Okay, so now what will happen to this chylomicron? So far we have known about the metabolism of the chylomicron, now about the degradation part. So this chylomicron remnant will now again go to the liver. Okay, what it does in meanwhile is that it will give all the apoprotein C2, AV, E, not E, C2 and AV back to your HDL. Okay, if possible, if it is still in circulation, all this apoprotein will be exchanged back to your HDL. With only ApoE, it will now enter into your liver. So the apoproteins that it has would be ApoB45 and ApoE. These two, it enters into the liver. Inside the liver, what happens? Because of the presence of ApoE, okay? Because of the presence of ApoE, it gets internalized, okay? Not through the LDL receptor, but by acting on the LDL receptor co-receptor. Because LDL co-receptor will get activated by your ApoE. LDL receptor will get activated only by your ApoB100, not by ApoB48. Okay, ApoB48 will just be seen as a label to show that it is coming from your gut towards your liver, but it doesn't help in the degradation of the chylomicron. For a chylomicron to get degraded, ApoE should be there. Okay, and it is taken by the LDL co-receptor. Okay, and it is internalized into the liver. Once inside the liver, it would be again degraded, and all the remainder cholesterol, triglycerides would be used in further metabolisms. Okay, so that we will know later. So what it does, what are the further pathways by which the chylomicron remnants would be utilized? Those are the salvage pathways sort of features. So what you can see further is that it can be used in bile acid synthesis. Okay, it can be used in the cell membrane formation. For even that purpose, it would be used. It's to a certain extent in the detoxification purpose also. It is using whatever cholesterol ester, cholesterol is there, triglycerides. It accumulates everything and it uses in the its own missionary formation, which is nothing but VLDL. Okay, so in VLDL formation, it will utilize all these proteins. So this is how a chylomicron undergoes its own fate. So now coming to this, now it would be easier for you. So the chylomicron's primary source would be intestine, and the density wise it is very, very, very lesser. Diameter wise it's very, very bigger. That's because of the high load of fat. It will not clump together. Okay, and molecular weight also would be lesser. And these are all the apoprotein that a chylomicron will have. If you look clearly, you could see ApoB48. Main is ApoB48, ApoC2, ApoE, ApoAV. Okay, AV or A14. So these are all the main apoproteins that a chylomicron will have. Now, once it becomes a remnant, you could see it is having only two chylomicron. One is B48, the other one is E. Okay. So we will move on to the next fate. you will see how VLDL, IDL, and LDL is now formed. So going on to the basics, your liver is now taking up all the cholesterol which is coming from the exterior, that is the chylomicron, and now liver will start to synthesize its own lipoprotein, and that lipoprotein is VLDL. Okay, so just like how an enterocyte is packing up a chylomicron, the liver will pack up a lipoprotein which is VLDL. So this VLDL will also have certain amount of triglycerides and cholesterol esters. Usually the triglyceride will be higher compared to a cholesterol ester. For example, if the triglyceride to cholesterol ester ratio in chylomicron is cholesterol ester to triglyceride in chylomicron will be in the range of 1:8 approximately. Whereas in VLDL, this ratio would be still higher, but it will be like 1:5, not like chylomicron, but at least to that extent it would be there. Very, very, very bigger amount of cholesterol esters would also be there. It is when you compare triglycerides, the cholesterol esters would be usually hydrophilic. They would be more of a neutral sort of lipid. Okay. So once this is packed, it labels it with apoprotein called as B100. And now this VLDL is pushed into the circulation from the liver. Okay. The VLDL will also undergo the same level of fate, like how your chylomicron is undergoing. Okay. So within the venous plexus, what it has, as usual, your donor will come, your HDL. This fellow will come, and this fellow will donate all his things. So he will donate ApoE, he will donate ApoC2, he will donate ApoAV. Okay. To your nascent naive VLDL. Okay. Once they receive that, they form mature VLDL. So it has now C2, AV, E, B100. No. It gets the attention of the main enzyme called as lipoprotein lipase. Okay. So because of the enzyme lipoprotein lipase, it gets hydrolyzed. And a certain level of triglyceride is still released. But these triglyceride levels will not be as abundant as in chylomicrons. Okay. Yet another important thing is that they give away these triglyceride and they form IDL. That is called as intermediate density lipoprotein. Okay. And to a certain extent, what happens with the help of the enzyme CETP, which is called as cholesterol ester transfer protein, some amount of transfer will also happen between your VLDL and your HDL. Okay. So your HDL also comes into pictures here. And with the help of this cholesterol ester transport protein, the HDL will transfer some level of its own cholesterol to your VLDL, and VLDL will give away its triglycerides to your HDL. So triglyceride, cholesterol intaking. Okay, so that happens, and that also results in the formation of your IDL. So IDL is actually something which is intermediate density lipoprotein. So this IDL, what it does is that it will enter back into the liver and it will have the same pathway, same fate, same pathway, like how your chylomicron has undergone. But this time, it is not the ApoE, it is the ApoB100, which will stimulate your LDL receptor. And with the help of the LDL receptor, it gets internalized. And inside the liver, on your IDL, there is a special enzyme which is going to act. And that enzyme is called as hepatic lipase. That is there inside the liver. So what does hepatic lipase do? The same sort of action which your lipoprotein lipase does. But lipoprotein lipase is outside your body, it's outside your liver, inside the vessels. But hepatic lipase is right there inside the liver. So it will break down all the remnant particles which are there in your IDL. So starting from cholesterol esters, it concentrates more on cholesterol esters and breaks it down. And also to a certain level of the lipolysis with triglycerides also happens. So finally, they degrade and they again help in the formation of VLDL formation. In this process, during the circulatory time, If this ideal is getting acted upon by either LPL or by the CETP to a certain extent, LDL can be formed. LDL is actually not the normal way of formation. It is just an intermediate pathway. Okay, usually the LDL would be not formed as a normal pathway. Okay, only when VLDL is very higher and thereby ideal gets very higher, the LDL will also get higher. Okay, that's because your ideal is now not able to enter into the liver. It is still present in your blood, and that is why it is getting converted to more and more of your LDL particles. So it is not in the routine fate of all these lipoproteins. It's just an accessory. So that is how LDL gets formed. Okay, so LDL also gets formed to a certain extent. Sometimes this can happen in your liver also. Okay, to a certain extent, the hepatic lipase it undergoes digestion. Some particles escape as LDL. Usually, it doesn't happen. It mostly happens in your blood. So this is the routine fate of your VLDL, IDL, and LDL. Okay, so what it does generally, you will have to know. At one note, we have seen it is taking all the food-related fat and delivering it to the tissues. Okay. On the other side, we can see what were present in the liver. The liver is synthesizing, and the liver is pushing all these things, and it is delivering it to the tissue. So this is what happens in the routine cholesterol transport. Okay. So you will see about individual lipases also in coming sessions. But at least to have a very simple understanding, you will have to know about this. HDL metabolism will come at the last, but with each and every metabolism, if you are learning the disorder, it would be even more easier for you. So if you come closer, if you look into VLDL, it is coming from the liver. Very, very rarely it comes from the intestine. Mostly it's coming from the liver. You could see the density slowly increasing and their diameter slowly reducing. And if you look into the structural components, it is most of lipid and only 7% it will have protein. You look into the apoproteins. These apoproteins are B100, same C2, same E. And ApoAV is not there. Sorry for that. ApoAV is not here. Okay. And the same thing will be there in ideal. Okay, the same thing will be there in ideal, and in ideal you could see the same thing. But when it goes to the liver, it will lose all these particles, and LDL will have only B100. Okay, so this is the fate of VLDL, IDL, and LDL. So this is the picture which is actually given in your Harrison textbook. This is a very simple picture depicting what will happen. You could see the dietary lipid being acted upon by the bile acids, and finally within the liver, within the intestine, they assemble and they form